Hey everyone, and welcome to the internet. So what we're going to be doing today is having a look at osmosis. So hopefully by the end of this lesson you're going to be able to describe this process of osmosis and when it happens. To start us off then, what we've got are 3 questions based around our previous lesson on diffusion. So have a go at answering those for me in your books. I'll just give you about 3 minutes to have a go at that. Okay then, let's see how we've done. As usual, if you've got it right, give yourself a tick. If you need to correct anything, then make sure you do so. So the first one, definition for diffusion. This should say the net movement of particles from an area of high concentration to an area of low concentration. So if you've got that right, give yourself a tick. The second one, we're looking for oxygen here. So it's oxygen that's diffusing into the red blood cells at the alveoli. Give yourself a tick for oxygen if you've got it. And then if we're looking at ways that we can increase the rate of diffusion for number 3, there are 4 things we could have put down. You can give yourself a tick for each one of these you got. So you could decrease the distance, increase the surface area, increase the concentration gradient, or increase the temperature. So add those up for me and give me a total score over in the chat window. Let me know how you've done. What we're going to be doing today then is having a look at another very important process in living things called osmosis. Now the first thing we actually need to know then is the definition for osmosis. So what I want you to do is write down this definition in that top box for me. So osmosis is the net movement of water molecules from an area of high water potential to an area of lower water potential across a selectively permeable membrane. Now, there's a few key things that we've got to make sure of in this definition when we write it. First of all, we need to be talking about the movement of water molecules. So osmosis is only to do with the movement of water. Secondly, we need to talk about it going from high water potential to a lower water potential, which we could also describe as moving down a concentration gradient. And the final bit is that phrase selectively permeable membrane. So once you've got that definition written down, what I want you to do is see if you can work out and write in your books what selectively permeable actually means. So think about what selectively means and think about the word permeable. What does that mean? Write me an answer to the question in your books after you've got the definition. I'll give you another minute to get those two things done for me. Now, in our definition, I did use a phrase that I think we need to clarify so we all know exactly what I'm talking about. And that phrase is water potential. Now, when we're talking about the water potential, what we're referring to is the concentration of free water molecules. Now, hopefully the two pictures I've given you there are going to help to explain this a little bit better. So what we've got in the picture on the left is a low water potential. So the big yellow blob in the middle is going to be a solute. It could be sugar, it could be urea, it could be anything, okay? And the little blue ones, they are water. Now what we can actually see is that lots of those water molecules, they've actually been attracted to the yellow blob, okay, our solute. So that means the number of free water molecules has been reduced. So what we've got there is a much lower concentration of free water molecules, so we've got a low water potential. Whereas if we look at the picture on the right, we can see all we've got are water molecules there, so they're all free water molecules, therefore that has a high water potential. So just make sure you understand what that word water potential means. It might help if you want to put those little drawings in just to help you to remember what we're talking about. So I'll just give you another 30 seconds in case you need it on this one. So what we can see in this animation is we've got small molecules able to cross that membrane in the middle, and they can do that because they are small. If we now change it to larger molecules like sugars and proteins, you can see they're not able to cross. What we're now going to do is have a look at water itself. Now, when we look at water, they're small enough to pass through the membrane freely. And this is what we're looking at with osmosis, the movement of water molecules. Those green ones we've just added in, they are urea, okay? Now, what we're going to see there is urea's too big to cross that membrane. But if we look, we can see that the water has been attracted to the urea, which has now reduced the amount of free water molecules on the right-hand side of our beaker. So what we now have is this net movement of water molecules from the left to the right, because what we're doing is they're moving down the concentration gradient. As a result of that, more water molecules are moving to the right hand side, the water level has gone up. Now, if we've got those two solutions with equal concentrations, we have something called an isotonic solution. If we've got one where we've got a higher concentration of solutes, so on the right, that is known as hypertonic, whereas one with a lower concentration of solutes is hypotonic. What I'd like you to do then in your books is have a go at answering this question. What will happen to the rate of osmosis when the difference in water potential is greater? So 30 seconds, write me an answer in your books. Hopefully what we've got written in our books is that the greater the difference in water potential, the faster the rate of osmosis. Now hopefully you worked that out. Because osmosis is just a special type of diffusion, then the same principles apply for increasing the rate. So if we've got a big difference in the water potentials on each side of that membrane, then osmosis is going to be happening at a faster rate. What we need to move on to next then is understanding what happens with osmosis in different types of cell. So we need to understand what happens in plant cells and what happens in animal cells. So what I've given you on the screen at the moment is a diagram that summarises what's happening in the plant cells. So what we can see is at the very top we've got a regular plant cell. Now those three coming off it at the bottom are showing us the different situations. So if we start off with the one in the middle, then what we've got here is our plant cell being placed into conditions that have got the same concentration in the surroundings as is found inside the cell. So this is what we refer to as isotonic. Now in that situation, because the concentration of the solution around it is the same as the concentration inside the cell, then there is no net movement of water. So that means the cell will remain the same. So if we were doing an experiment on this, it would remain the same length, the same width, the same mass. It shouldn't change. If we now go to the diagram on the left hand side, then this is where the surroundings of our cell are at a lower concentration or higher water potential than the contents of the cell. Now in that situation, what we've got is a higher water potential in the solution around it than inside. So the water moves from the high to the low water potential. It's moving into the cell itself. So what we see as a result of that is the water moves into the cell by osmosis, and then it's going to make it expand. Now hopefully we remember plant cells do have a cell wall around the outside, so the vacuole is going to expand and push out against the cell wall. And when it does that, it's going to create something called turgor pressure. Now it's that turgor pressure that makes the cell become what we refer to as turgid. So it keeps it structurally solid, if you like, and allows the plants then to be nice and vertical without the need of a skeleton. If we go to the diagram on the far right, then what we can see is the situation when the surroundings of the cell are a lower water potential than the contents. So in this scenario, the higher water potential is inside the cell, and therefore water is going to move from the cell to the surroundings. Now what we see as a result of that is turgor pressure is going to fall and the cell becomes flaccid. Now eventually, if this continues, then actually what we see is the cell basically collapsing in on itself, and this is a process called plasmolysis. So what we see is the cell membrane pulls away from the cell wall and it all just shrivels up in this plasmolysed cell. So what we've got there are the three different scenarios we could see. Now, as we said, with the isotonic, okay, the one in the middle, then those plant cells we've put in that solution that's the same concentration inside as it is in the surroundings, they just don't change. If we place it in a solution that's got a higher water potential than the cell contents, the water moves in. So those cells are going to expand, and if we were measuring it, it would get longer, wider, the mass would increase. And the opposite would happen if we placed it in a lower water potential solution. Because the water is moving out of those cells, what we see is the mass decreases or it shrinks a bit. So what I want you to do now then is just explain what happens when the plant cell is placed in those different solutions. So I'm going to give you about 4 minutes to get that done. If you want to include diagrams, by all means feel free to include any diagrams to help you. So ordinarily what we'd be doing at this point is the osmosis experiment. It is one of the assessed practical tasks, And what I've actually done is on the website, I've put some instructions for you to be able to do this in very simple terms at home. But one thing that we do need to understand is that when we're looking at these changes, we should always calculate the percentage change. And hopefully to work out percentage change, we've done this in maths, I would hope, it's your final mass minus the initial mass divided by your starting mass or your initial mass, and then because it is a percentage, we times it by 100. So head on over to the website, to the resources, to see how to do a little experiment at home. Okay, so the second type of cell we need to understand is the animal cell. Now, hopefully we remember from our work in B1 that an animal cell has no cell wall. So it's got a cell membrane, but no cell wall. What we're going to do is, I've given you the pictures, same as before with the plant cell, but this time we've got our animal cell, a red blood cell in this case. Now, what we've actually got, we'll start with the middle picture again, is if we place it into an isotonic solution, so the surroundings have the same concentration as the cell contents, then what we find is there's no net movement of water. So the cell remains unchanged. The same amount of water moves in as moves out, so there is no change to our cell. If we look at placing our red blood cells into a solution that has a higher water potential than the cell contents, then water is going to move into that cell, but this time it's going to swell up and eventually it will burst. And this is a process called lysis, the bursting or the splitting of cells. So what we actually see there is, because there is no cell wall, then our red blood cells will literally fill up with water until they're at that breaking point, and then they burst in that process called lysis. If we place our red blood cells into a lower water potential solution than the cell contents, then the water moves out of the red blood cell and it shrivels, okay? It's going to shrivel up into this type of cell called a crenated cell. So again, what I'd like you to do is explain what happens when we place the animal cell into those three different solutions. You can include the diagrams if you want, and do make sure you are using those proper terms, lysis, crenation, and obviously explaining what's happening and why. So just to finish off for today then, here's our plenary questions. So those of you working up to a grade 5, it's the 2 on the left. For those of you working over a grade 5, it's the 2 on the right. Don't forget, you can find the answers to the plenary questions over on my website, and the link is on the screen there for you. You can also find some additional work and also those instructions to the little investigation that you can have a go at home. So head on over to the website to get those extra bits. Because it's Easter next week, then we will be back on Wednesday the 15th of April with your next combined science lesson. So see you then.